Nice and nasty about why people act nicely or not so nicely. And Charlie has pitched to me the idea for their next book, which is The World of the Elderly, to kind of wrap it all up. Okay, but you want to know more about Daphne the person. So one insightful example is years after I left her lab, I said, Daphne, one of my favorite things that you taught me is how to take negative feedback. Right? I told her that when I received the rejection letter from the editor, my reaction was anger, right? Sadness. It's the worst day of my life. But you said, Mayu, if we take all of the weaknesses identified by reviewer 2 and we address them, don't you think this manuscript will be that much stronger? And of course, she was right. So I said, thank you for giving me that insight. And I thought she was going to say, yes, Mayu, finally you have learned one of life's great lessons. Instead, she said, oh, thank God that was your perception, because I was really shitting my pants. Maybe she didn't use exactly those words, but that is how I recall the conversation. So I think a great mentor, a great leader, is someone who will put on a brave face for you, but when the time's right, will also tell you the truth. So I think today she's going to tell us a little bit about the truth behind being barely tenured to Officer of the Order of Canada, Dr. Daphne Maurer.
Thank you so much. Is the audio working? Everything okay? All right, back row says it is. So I arrived at McMaster in 1973 with the data for my PhD thesis, which was not quite done. I was recording eye movements in infants. There were no automatic eye trackers at that time. In order to record babies' eye movements, we filmed them with reflections on their pupil, and then we measured the location of the center of the pupil and each of the reflections and wrote programs to translate that into where they were looking on the stimulus. It was very slow. Laborious, frame by frame scoring. But it allowed us to find out some basic things about early perception. Can babies see straight ahead? Can they see off to the side? How do they scan faces? I was doing those studies. I finished my PhD. I built a lab in the hospital next door for studying newborns, a lab in the basement of this building to study older babies, recruited 3 graduate students, and we went to work. Now, the context of this is I was the only developmental psychologist in the department. And that was true for the next 19 years until Laurel Trainer was hired. So the work I was doing was slow, not only because it was laborious, because babies sleep. They come in for your experiment and they fall asleep and you get 0 data. All the methods I was using required 2 people in order to test a baby. It took me 1 year to get a data point. I had colleagues doing really good work in cognition and perception, one of the big strengths of this department at that time and to this day, who could have an idea and get a data point in a week. So the context was very different between what I was doing and what they were doing. So by my 6th year, I did not get early tenure. I was evaluated, but there was no hope. There was just not enough productivity to get early tenure. My 6th year, the crunch year, you get it now, or thank you, Daphne, goodbye. I had a good grant from NSERC, I had a methodological chapter, and I had 4 papers that were either published or accepted, and I just squeezed through getting tenure. And then something happened that allowed me to speed up the work, not to the level of cognition or perception, but much, much faster. a method was described in the literature where all you needed to figure out was, is the baby looking at the stimulus or not looking at the stimulus? You didn't need to know whether they were looking, in this case, at the eyes or the mouth, but just on the face or off the face. It was called an infant control procedure invented by Les Cohen, and it was really important because there are huge individual differences in babies. So if I present that stimulus to a 3-month-old, as is happening here, some babies will look for 5 seconds, and some babies will look for 3 minutes. So if you have fixed duration trials, you're really messing up what you're measuring. So we started using infant control trials, presenting stimuli to babies, and using another trick. We would present the same stimulus over and over and over again. And eventually the looking times go down. They're not 3 minutes anymore until the baby was obviously habituated. And the criterion we used, that their looking time had gone down 50%. 50% relative to the they started, not to a group average. That also was a new trick at the time. And then we would give a test. One trial of the test would have exactly the same thing, and the baby wouldn't look very long, and then we would switch to a novel stimulus, in this case, a novel person's face. And babies often showed us in many ways they could make that discrimination by looking longer at the new one than at the old one. So we started running through a lot of basic perceptual problems with babies using this technique. We tested the ability to discriminate between 2 different orientations, and 1-month-olds showed us they could do that. That was really important because at the time there was a hypothesis that the visual cortex is not working at all in the first few months of life. And we argued from this orientation discrimination that it probably is, that probably is cortically based, although you can stand on your head and come up with a few very arcane other hypotheses. But it was an important paper for that reason. We tested babies' ability to tell the difference between a schematic drawing of face and scrambled versions and showed that 2-month-olds could do that. We tested the discrimination of facial expressions, check at 3 months, and we tested the ability to discriminate between 2 women's faces. So it was producing faster. This work was very easy to publish because in every case, I was showing that babies could do something at a much younger age than had ever been shown before. I got promoted to professor on my 2nd try. I failed to get an on-time promotion at 10 to 12 years. And when that happens, the department recommended it. It was no problem, but it failed somewhere, faculty or Senate. Senate had to write me a letter telling me all the things that were wrong with my career that caused me not to get tenure. So I got a 2-page letter of criticism. It hurts thinking about it to this day. And what it boiled down to, it seemed, was negative comments, reserved comments from reviewers. So one reviewer had said she's doing really good work, but it's scattered. She hasn't concentrated on baby's color vision or baby's discrimination of orientation so that she has a body of work, so that she's known internationally as the person who studies that. So in my mind, it was all connected. I was trying to characterize the perceptual world at the beginning and draw inferences about what that meant about the brain at the beginning. But to the reviewers, that didn't come across. I came up again, I chose to wait 2 years. I came up again, and I recommended a different kind of reviewer. Now, when you come up for these reviews, the department asks you for recommendations for reviewers. They don't have to use them, but remember, I was the only developmental psychologist in the department. Nobody else knew anybody to ask, so I'm pretty sure they probably used the ones I recommended. The first time when I failed, I had chosen rising stars, people just a little bit ahead of me. Second time I said, it might be better to choose really senior scholars who have more perspective about contributions to the field. I did that at cell 3, all the way up, and I got promotion. You know, of course, it was 2 years later, I had more publications. We don't know that that's the reason, but I have a suspicion. And I've been on other people's tenure and promotion cases. My advice to my young colleagues is choose senior people to be your reviewers. Okay, then something really awful happened. I submitted this paper for publication. I'd already published one showing that babies could recognize the mother's face at 3 months of age. Didn't know about fathers. We did the same study, same methodology, and it was rejected. And the principal reviewer said, okay, she's published mother's face. Now we get father's face. What's next? Cousins? Grandmother? The family dog? And I realized that what I had been doing was not really developmental research. What I had been doing is showing that there's a super baby who can do this at 1 month and that at 3 months. But I wasn't studying development. I wasn't studying why an ability changes from this into that. It was essentially anti-developmental work. I really needed to change direction. I needed to understand what's going on that is driving this development. And just at that moment, I got a chance to study the role of early experience in driving these postnatal changes. I got a call from the hospital for sick children saying, would you come in and do our eye clinic rounds at 7:30 on some date? And I said, sure. Who's the audience? What do you want to know? Why are you inviting me? And they said, we want to know how to test baby's vision. Okay, why? Because we have all of these babies who were born with congenital cataracts. We're doing the surgery. We're fitting them with contact lenses, and the lenses fit. We're good at that. They don't slip. They don't irritate the eye. But we have no idea if the babies can see or what they can see. So here's a kid has congenital cataracts. That's the white in the middle of the lens. That means that the patterning in light is not making it undistorted to the retina. And you know that because an ophthalmologist looking into that eye can't even see the retina. So visual experience is being blocked in this eye. There's surgery to remove that cataract. The child is fit. That means you're taking the lens out of the eye, and the child's given a compensatory contact lens. Well, as a developmental psychologist, I had no idea that there were large numbers of such cases. I knew the occasional case study in the literature. Like, this is gold. These are kids who miss visual experience for a short period of time, and then they get it, and we can look and see how they're different from babies who always had normal eyes. So I ended up collaborating with ophthalmologists at the hospital for sick children. Fortunately, they saw all of these patients on Thursdays. That's when they ran the contact lens clinic. So for a long, I think 20 years, 4 of us, including Terry Lewis and 2 RAs, took the train to Toronto very early every Thursday, dug our equipment out of the closet, set it up. We needed 2 people for each test. We're doing a whole bunch of stuff. And as the patients, we had the techniques for babies. As the patients grew up, we developed techniques for testing toddlers and then young children and old children. And I'm still studying them. Some of them are age 30 now, 30 years. Now this work was slow too. There were only about 10 new patients a year, and we studied them longitudinally. So the data came in slowly. But it was unique. Any child in Ontario got referred to one place. So we had the largest cohort in the world of such patients. And it was not easy, it was not hard to convince people of the importance of the work. It was easy to publish, it was easy to get funded. I ended up with funding from the March of Dimes, from SSHRC, from CHR, from NIH. We got papers in Science, Nature Neuroscience, Current Biology. I built a much larger lab, and things flourished. And a line of work that I'm known for. So my lesson here is keep your eyes open. Ask questions, grab opportunities, dare to change direction. You know, I was doing totally new stuff, but it certainly paid off. Now, what were we doing? So here are the patients. They start with cataracts, no patterned input. They have surgery. Surgery was as early as 1 month or as late as 9 months. After the eye heals, they came back to the contact lens to get that contact lens, and then we started measuring their acuity longitudinally from the moment they could first see. We got data within 10 minutes of their first being able to see. We've studied about 20 aspects of vision in this cohort, and only 4 have not been impaired, and they are here. The patients appear to have normal color vision, normal ability to discriminate shapes, whether it's the shape of an eye in the face or a basic geometric shape, to detect quite accurately biological motion, and to detect whether something is a face or not. And interestingly, all of these are present at birth in at least a crude form. So all of these have a neural underlying architecture that can develop in the womb in the absence of visual input. And they're preserved even if you have a fairly short period of visual deprivation. So we can say that the baby comes biologically prepared with certain rudimentary skills, and we found all of those were normal. But the list that's going to go under the next column, which is deficits, is much longer. So let's look at acuity. So acuity in children with normal eyes, this is the developmental trajectory. So it's tested with grading, because babies like to look at something patterned like stripes, and you can test them with really wide stripes, and then you can make the stripes smaller and smaller, and determine what are the smallest stripes when they'll look towards the stripes instead of a plain gray. If you're an adult with normal vision, you're up there, a little bit better than 1 minute of arc. Newborns are down here, about 60 times worse. They're not blind, but they're about 60 times worse than adults with normal vision. And then there's very rapid development in the first 6 months, and then just a very, very, very slow progression out to about 7 years. Well, when we tested babies, the moment they could first see, all of the values fell within this ellipse. They were at the value of a normal newborn. There had been no acuity development in the absence of visual input. And that meant, of course, the child treated later was farther behind than the child who was treated early. But there was rapid recovery. So by the time they got to the first birthday, they were all normal. So, you know, this kid who's way, way, way behind, there's an accelerating curve to normal. So the system was ready, expectant, ready to respond to visual input, and that impulse was increasing in strength even during the period of visual deprivation. The ophthalmologist relaxed, everything's working, but we kept testing. And when the child got out to 2 years of age, they hit an asymptote. They stopped improving. So their acuity, at 18 years, their acuity now at 30 years is like that of a normal 2-year-old. So, and we found deficits In all like that, in all of these things that I've listed here, Manu, by the way, did the work on facial identity that's there. And it's a bit puzzling because babies sleep a lot, as I complained about a moment ago. They don't see very well. I mean, why does it matter so much if you miss a little bit at the beginning? Because it wasn't that great input for the child with normal eyes. And so what we have come to realize is that early visual input is setting up the neural architecture that allows refinement later. If you don't get the visual input, later you don't have the optimal neural architecture to have all of those skills at the full level. So this cartoon captures it. You know, you have to buy it, the candy house, so you can tear it down and build a 44-story building. And we've come to call these sleeper effects. So there's an effect of early deprivation. It's only manifests much later in life when you fail to develop a skill. So what's happening during this early period of time, the biologically prepared organism is tuning in or adapting to the environment. So it's being refined, it's being tuned to the environment, and that involves pruning of connections that are rarely used and inhibition of the ones that remain that don't match the environment very often. And that is failing to happen in these cataract patients. Okay. Next, I want to talk a bit about a detour I took. And this arose after Charlie and I wrote that book, The World of the Newborn, because it was a neat way to find out more about exactly what's that biological preparation like. Because in this book, we said, oh my goodness, all of this conversion evidence suggests that the newborn has synesthesia. Okay, so what is synesthesia? So in adults with synesthesia, it comes from the roots for union of sensation. They have extra percepts. There are 63 or more different types. A particular note is idiosyncratic. So for one synesthete, might evoke the perception of the color blue. For another synesthete, might evoke a star. And for another, oranges, but oranges that aren't quite ripe. So the associations are very specific, but they're idiosyncratic. What we know from studies of synesthesia, it's automatic. People aren't thinking about it. They, in fact, have a great deal of trouble suppressing it. It's consistent across the lifetime. You can test a synesthete 10 years apart, and you'll get the same answers. It runs in families, so there is a genetic component. And if you count all of the types, it occurs in 15% of the population. So I'm sure there are people with synesthesia sitting in this auditorium at the moment. Now, what seems to be happening in synesthesia is there's less tuning into the environment. There are extra neural connections that don't get pruned away to match the environment, and that don't get inhibited even though they don't match the environment, and that's the origin of synesthesia. So we said, hey, wait a minute. There might be some similarity between the baby who hasn't done the pruning and inhibition yet and the adult with synesthesia. Here's a whole new source of ideas about what's going on in infant perception. And vice versa. What we know about infants was a new source of ideas about what's going on in synesthesia. And so we started to test those ideas. I'm going to give you one example. So large-scale studies of English-speaking synesthetes show this agreement. One of the most common forms of synesthesia is to have colored letters. So in English-speaking synesthetes, A's are red, B's are blue, C's are yellow, etc. at levels exceeding chance. You always have to take chance into consideration, but there's quite a bit of agreement. So why should that be? Well, some of them are clearly based on the language. B is blue, it's the first letter of that word. Why are I's white? Why are O's white? Why are C's yellow? Some of them it's not quite so obvious. So we decided to test toddlers who don't know the alphabet. And even if they know a few letters, they don't know that B is the first letter of the word blue. We developed a hiding game where we had transparent versions of a letter. We had to be very careful. There was no color in them. We had to find the right plexiglass to make them transparent. And then we played a game that says, my friend the X, this is my friend the X. My friend the X is hiding on one side of this black or white box. There was always two choices, because these are 2.5-year-olds. Where do you think my friend is hiding? And they would choose, and they always were correct. We always found that, I was indeed hiding there. So we did this with 2.5-year-olds, and for comparison, we tested 7 to 9-year-olds, and because they can read, and we tested adults. So when we tested, A's are red and G's are green, toddlers are random. So this is the percentage choosing as we expected, this way. School children got it, and adults got it. These are ones without synesthesia. And so that was consistent with our hypothesis. A's are red maybe because A is for apple and apples are red, or research since then suggests that the first letter of the alphabet matches the first color word the child learns. And in English, that means A's are red. In other languages, it's the first letter of the alphabet goes to the first color word the child has learned. Okay, so these associations we found in English-speaking synesthetes are like that, not in toddlers, school children, and adults. So this changes with reading. They emerge in Anglophone synesthetes. They seem to be learned associations. But look what happened with X's are black and O's are white. Toddlers got it. They were just as accurate as school children and adults, and searching in the black box for X, in the white box for O. And that is the pattern that we found for all of these. I is O, I is white, O is white, those are black, and C's are yellow. We found that we knew it was true in synesthetes, and more recent research shows it's true for synesthetes speaking English, Dutch, Greek, Korean, Spanish, and Russian. It's true for toddlers, it's true for school children, it's true for adults. So we call them natural associations. Developmental psychologists don't like the word innate, so I just call them natural. There's no obvious learning signal, but they seem to reflect something about the intrinsic organization of the brain. Okay, so the moral here is read widely. Go to colloquia, way outside your specialty. Sometimes investigate a wild idea after you have tenure. And while you're still doing your traditional research on the side, if you have a solid argument, you can even get funded. I got funded by NSERC to do the synesthesia research. So it was wild, but I had a good argument and I could get funding for it. Okay. I retired in 2012. I continue to do the research with the cataract patients. They're still volunteering. They're between 30 and 40 years old. But I returned to kindergarten after I retired because I discovered that Ontario had stopped doing vision screening in schools in the 1970s. Now, why did I care? Because 5% of 5-year-olds have amblyopia. They have two eyes that aren't working together because one is turning in or they have different refractive errors. And if untreated before about age 8, that is a lifetime loss of vision in one eye. Somewhere between 3 and 17% have astigmatism, which means that this picture on the left, if it's 3 diopters, would look like that on the right. It's 3 to 17% because the percentage that's found in studies varies a lot with location, with social class, with ethnicity. Even if it's only 3%, that's a big chunk of children. And between 5 and 26%, again, quite a range because of geographic variability and ethnic variability, are farsighted. So when they're looking at text, the text on the top line in front of them, depending on how farsighted they are, it would look like the second line or the third line. So in collaboration with an ophthalmologist, the Hospital for Sick Children, we set out to do something about this. We needed to figure out how to do effective vision screening. We started in 2014 and convince the government to implement whatever we found was effective. So we did a whole bunch of studies. I'll very briefly mention one of them. This is one that Maurer was the research director for. So we would go into the school, test the children with the 5 best evidence-based tools in 2014. Acuity, stereo vision, 2 measures of refractive error, and a measure of binocular vision. And if the child failed screening, we brought an entire optometry clinic into the school, and the child got a full optometry exam, and if they needed glasses, they were provided at no charge. I just said so we had to buy all the equipment across the top. This is what it means to bring a full optometry clinic into a school. We had to buy all of that equipment, and we were working at such a pace that we duplicated it because we wanted to be able to be in 2 different parts of the province at the same time. It's all the normal pieces. When you go to your optometrist, they're all there. We went to all of the communities that are listed at the bottom along with their population, and 52 schools in those communities. That means we collaborated with all of these organizations. There were a huge number of permissions and approvals to get. We also needed to find optometrists willing to work in those 52 schools. So we needed big bucks. And I've learned that applied research is really quite tricky. It's got a lot of steps, and it's not so easily funded, because when we applied for funding, reviewers said, what's the point? We know vision screening is valuable. Why do you need to do a study? Or they said, we already figured this out in the United States and published it in the year 2000. Why do you need to do a new study? Well, this is 2016 by this time, new tools are available, and we have a totally different medical system. But getting funding was tough. So I learned that applied work has its own challenges. I would save it until you have extra time on your hands. But it's important, and it's better than just grumbling and complaining, like, we don't do this thing that we should do. Well, you can do something about it, and you can make a difference, because we did manage to change the public health standards for Ontario, so that now every public health unit is required to either do or to make sure there's vision screening of senior kindergarten children in every school. And the tools that are recommended, the cutoffs, the choice of age, was based largely on the data that we had obtained in the previous 3 years. I have just a couple more slides, a couple more lessons. Collaborate whenever you get the chance. You get new perspective, you get new ideas. I was fortunate to be part of a Human Frontiers grant. It involved 4 labs in 4 different countries. I wrote and got a James S. McDonald Foundation grant, which we called Brain CPR. It was all about, can an adult brain recover from an earlier insult? We had 5 labs doing research both on humans and animals, doing both experimental and clinical work, and we were in 3 different countries. Whoops. And take on leadership positions. These are the main ones I did. You know, being on NSERC, being president of the Faculty Association, chairing the ethics board, being on the national board that sets the policy, being president of the Maine Society for Studies of Infants, and recently, I'll come to this one in a minute, being a member of National Academy of Science expert panel. Yeah, it's service, but it's also fun, and you meet other people who are interesting outside psychology often, who have ideas that you can work with. You learn things about yourself. I discovered I have some leadership skills. I never knew that until I was president of the Faculty Association and had to figure out how to deal with 1,000 members with 1,200 opinions and the administration at the same time. And when I was president, we sued the university because what they were doing to our pension plan. So how do you remain on good terms with the administration you're suing? I know it was a good learning experience. I think I handled it reasonably well. And the opportunities keep coming. I retired in 2012, but a year ago I was asked to sit as an expert, as a member of an expert panel of the National Academy of Science, looking at the increase in myopia in children. Myopia is nearsightedness, it's going way up. At age 17 in Seoul, Korea, men who grew up in Seoul, they have to report for Army service and they get their vision tested. 96% are nearsighted. When I say it's going up, it's going way up. There are no data for Canada. For the United States, the last national data were collected in 2004. There are some regional data in the United States that also show it's going up. It's not at 96%, but it's going up. And it's serious because nearsightedness means your eyeball has grown too long. So as the body grows, the eyeball has to grow longer to keep a pace, but it needs to stop. If the stop signal doesn't work, you become nearsighted. And in a lot of kids, the stop signal isn't working now. That's really alarming. And they are going to be at increased risk of glaucoma, detached retina, all kinds of retinal problems because the longer eyeball is stretching the retina. So I got to work on this with 14 other people over the last year. Our report is being released on Tuesday. It's a 400-page report, and the literature was fascinating. It was fun to work on it. So, you know, if you volunteer, the opportunities will keep coming. And then one day, out of the blue, came an email. Can I have your phone number? I need to call you in the next 48 hours, someone I didn't know. I almost deleted it. He wanted to call me to say, you have been selected to become an officer of the Order of Canada. Will you accept? And of course I said yes. And then he said, okay, it's embargoed for 2 months. Don't tell anybody. So I'm going, I have the biggest dose of imposter syndrome right now that I have ever had in my whole life. I'm astonished. You don't know you were nominated, and then suddenly it happens. It feels wonderful and humbling at the same time. And this career, this is a wonderful job. You know, you essentially get to study whatever you're curious about and to train students how to do that kind of research. And it's especially wonderful if you're an interactive collegial department like this one These were the working conditions that made all of this possible. So thank you, PNB, for making possible what I just told you about. Thank you. And I've been asked to put up this slide. I'm giving a completely different talk on October the 9th about that third book Charlie and I wrote. Anyway, there's the announcement. Okay, I'll just leave it there.
Any questions?
Yeah, it was a really inspiring talk. I'm curious if you had any potential explanations for the synesthetic X black and O white.
Well, we've speculated, but it's pure speculation, that things that are continuous are perceived as white, I's and O's, and things that are discontinuous, like the X and the Z, are perceived as black. And the colors tend to be opponent in synesthesia, so, black and white, that's really basic. But I have no idea why C's are yellow.
Yeah, I was thinking the X black and the O white could be a Kiki Boba kind of thing of sharper, darker,
Right.
smoother, lighter. I don't know. But yeah, the yellow I didn't.
Yeah, we also did Kiki and Malumba and Bobo with the toddlers, and they think that the sound, Kiki, is jagged. And the Kiki is going to be we did a hiding game, it's going to be hiding in the box that has a jagged shape on the top. And Booma or Malumba is going to be hiding in the one that has a amoeboid shape on the top. So all of these things may be connected, but I don't have a hypothesis about why C's are yellow. And after that, the ones we tested, everything failed. And at some point you quit when everything failed.
If it wouldn't scoop your report, can you make any comment about why, with nearsightedness, why nearsightedness is now almost universal among these Japanese Korean men? Because that's not just a rise in incidence, that's a major transformation.
Well, the report's embargoed until Tuesday, but I've read the literature, so I can tell you what the literature shows. There might be an effect of near work, because the more near work you do, the more likely you are to be nearsighted. So it turns out that the more years of education you get, the more likely you are to be nearsighted. 2 more years of education, half a diopter more nearsighted. But what we have really good evidence for is the stop signal's not working. The eye keeps growing. The stop signal's not working. Part of the trigger to stop comes from something that happens when you're outside. There is a difference in luminance between sunlight and inside that's 100 to 1 or 1,000 to 1. I mean, it's huge. And we have retinal ganglion cells in the retina that respond to light levels. They don't do contrast, they don't do patterning, they're related to our circadian rhythms, but it's also thought they're part of the circuitry for the stop signal. So we know that kids who don't get enough time outside are more likely to be nearsighted, and at this point there have been randomized controlled trials where you have this classroom go outside 3 times a week, and this classroom can stay in. These were done in Asia. To keep the kids outside, you have to lock the doors to keep them from coming in and studying. Anyway, going outside slowed down the progression of nearsightedness. So we know something about being outside matters. Whether there's an additional decrement from near work, we don't know. It's actually interesting because the cross-sectional studies show correlations. More near work, more likely to be nearsighted. Okay, slam dunk. But that's not causal. The longitudinal studies fail to find an influence in near work. So if we take 5-year-olds, none of whom are nearsighted, and we track them longitudinally, and we track their near work, and we look at who becomes nearsighted, the near work had no influence. So we don't know. And near work, reading a book most of us don't do it outside. So there's a confound there. Screens are probably not the answer. The increase started in the year 2000. Kids didn't have screens in years 2000. The analyses show that the correlation with screen time is identical to the correlation with near work. So if there's any effect of being on a screen, it's because it's near work. There's a little hint that maybe the answer might be different if you're a 2-year-old. And those studies haven't been done yet. Now, we know that being on a screen isn't worse than reading a book, once you're 6, but we really don't know in that early period. That research hasn't been done. Jason Hart.
I have a thesis student who is asking this question, and they were working on this, but they got a question.
Daphne, that was a fabulous talk. You gave us some things that we could learn about, and you also told us about a wonderful career. You told us about a lot of things that you were very glad you did in your career that kind of opened up doors and gave you opportunities. If you look back at your career, are there things that you wish you did differently?
Every day there are things I wish I did differently.
Are there things that you could advise us not to do?
Oh, that's a good question. I'm an optimist. I try to put things behind me. I never published the paper on the father's face. I was so devastated, it was no good. I didn't submit it to another journal. 30 years later, somebody published a study on the father's face. We didn't know it for 30 years. So, yeah, I probably regret that I didn't realize that you could submit it to a different journal. But that's not really an answer to your question. I was asked many times to chair the department. I always said no. I don't regret that. I always said no because I had really good relations with my colleagues and I didn't want to become their boss. I didn't want them to perceive that I was the boss because the relations shift. Inevitably, they shift. I just became president of a social arts and letters club. People treating me differently. It's like, it feels not good. Sorry, Mel, to say you should never be the partner. We've had fantastic chairs. They were very supportive. I'm glad they did it. And it's also a pretty thankless job, I think, because you deal with a lot of confidential matters. You can't even tell people what a good job you're doing. I'll have to think about that.
Any other questions? Yeah, Bruce?
So, you described a lot of decisions that you made in your career where it's remarkable. You talked about, it seems like you emphasize the big picture views, taking risks, taking on real big challenges. But I'm guessing that really worked for you, and you've supervised many graduate students as well. I imagine that the advice you give to graduate students isn't just to do exactly what you did. So I wonder if you could just unpack.
Okay, that's
the question I'm asking. It's like I'm just going to say, you must have some remarkable abilities to tackle those kinds of big challenges that hold high risks, and congratulations on that, those amazing skills. But I imagine you supervise people, and what you did maybe might not be the best advice for everyone.
I said after you get tenure, and things like that. I misled you. I was always doing bread and butter, surefire research at the same time. So this was, it was a considerable investment in time, but it was a sideline. And I would advise an incoming graduate student, we would explore what they were interested in. We would talk about risks. So Farron Spector worked on the synesthesia work, which was quite risky. She ended up with, I think, about 5 papers on synesthesia in kids or in adults. But you don't, and she will be here later, maybe later this term, in the celebration of, is it secret?
No, it's not.
Of Lewis being elected to the Royal Society of Canada and me for the same old thing. And, but she did it with her eyes open, and the studies, we could complete a study in maybe 6 months. It didn't take a year, and we were doing several of them at the same time. So you don't put all your eggs in a risky basket, and you're more careful before you're free after your last promotion, and you're more careful. Yeah, as you get more senior, you have to be quite as careful. Junior colleagues, I know, have sometimes run into the same sort of problem I did because they're doing a whole bunch of things, and none of them have gone far enough to be world famous at the time they come up for tenure, come up for promotion. But all of them look really interesting and really important. And I've helped a couple of them get through, but it was a struggle for them to get through. So, if you're doing that, I wouldn't say don't do that, because the reason to be a prof is to be able to explore your curiosity and do what you think is interesting. So I'm not going to tell anybody, if you've got 3 ideas, only study one of them because you're more likely to get tenure, because that's not you. You're not going to enjoy the job, but just do it with your eyes open.
I was like, it's like a similar question, but I was wondering. I'm also working with intense kids, and special population and data collection can be sometimes so stressful, and you don't get the data easily compared to other studies. So I was wondering, how did you deal with that frustration?
Yeah, so as a supervisor, I could be working with students on several different projects at the same time. As a graduate student, you're probably just working on one thing, or as an undergraduate thesis student, you're only working on one thing, so you're not quite in the same luxurious position. I repeatedly had to educate people about how slow developmental psychology is. Every time I wrote a grant proposal, there was a paragraph in there about the special problems of doing developmental psychology, and I imagine it's going to be the same in applied. It's probably the same in many forms of neuroscience. I mean, this is not unique to developmental psychology. You have to explain to your colleagues in the department, in the faculty, in the Senate, not when you send something in for review, and when you're applying for money, that it's slow. But it's so valuable, it needs to be done. And it's fun.
Yeah.
Yeah, I had a question about the color associations with ABC.
Yep,
You said you couldn't think of a reason for why C is yellow.
yep.
But you said red, like one of the reasons you thought of for red was that it's the first color that kids learn. So couldn't C be yellow because it's like it was red, blue, yellow, red. So the first 3 colors that kids would learn would be red, blue, yellow.
Cool. Okay. That's a good hypothesis. So bees are blue because they could be blue for either reason. Okay. Yeah. That's very clever.
Any other further observations? Okay, well, we have a reception in 2:05, so and continue the conversation. Oh, and Sally was a big help in all of those schools that we went to, carrying all of that and talking